In this video, we'll be doing a filter-based DNA purification. So at the beginning, our pellet contains our DNA, and the liquid is just waste, so we can just pipette this liquid off, being careful not to disturb the pellet because we don't want to lose DNA, because the liquid is a contaminant. Now that we've done this, our DNA is actually usually trapped in something like a gel, so we'll add lysis buffer to release it from this gel into the liquid that's contained in the buffer. The lysis buffer is pretty acidic, so after we've added the lysis buffer, we'll want to add a neutralization buffer in order to bring it back to a normal pH so we can proceed with the process without the solution being too acidic. After we've added the neutralization buffer, our pellet will just contain waste. And we'll actually want to use something called a filter tube that has two parts, a top filter and a bottom reservoir for liquid, and we will pipette our liquid containing DNA into the filter tube. And when we've done this, the bottom part, which has a pellet, is just waste. We want to be careful not to disturb it so that our DNA is pure and doesn't contain the contaminants in the pellet. And we can just get rid of the pellet at this point. Now when we centrifuge the filter tube, what it will do is it will pull through the liquid with contaminants through the tube, and the DNA will remain in the filter. So it's kind of backwards from what you might expect. The DNA is remaining in the filter, so we want what's on the filter in this case. Now that we've done this, we can purify it by adding something called wash buffer. And this wash buffer, when it's centrifuged, it'll pull contaminants off of the filter but leave the DNA on the filter so that the bottom reservoir will contain liquid with contaminants. So again, we'll centrifuge it, and the centrifuge is pulling the liquid down, so that's why we have to centrifuge to pull the liquid through the tube. When we've finished centrifuging, we will take the bottom part off of the tube because the bottom part has the liquid that is contaminants, and we'll pour this off into a waste beaker to make room for more liquid because we're going to want to wash it again to further purify the DNA because we want very pure DNA. And this time when we add wash buffer, it'll do the exact same thing as before, where the wash buffer will pull contaminants through the filter, but the DNA will remain on the filter. So again, when we've centrifuged, the liquid will just be contaminants, and our DNA will be on the filter. So again, we will pour off the liquid, which contains contaminants. And now that the DNA is pretty pure, we can just tap it a few times on a paper towel in order to dry it off and get off extra wash buffer. So we'll want to gently tap it. We'll get a new bottom half of a filter tube because this new bottom half needs to be free of contaminants. And now that it's purified, we're going to add something called an elution buffer. And what this elution buffer will do is first, when we let it sit, we'll usually let it sit for around 10 minutes, it will soak up the DNA into the elution buffer from the filter so that the DNA will now be in the liquid. And when we centrifuge it again, then the DNA this time will be pulled through the filter with the elution buffer and will be in the liquid. And that's our final product, is a liquid containing the DNA that we'll want to be sure we do not throw away. And then at this point, we can get rid of the filter and just keep the liquid.